So, hello everyone. So, today we are going to move ahead with the next session of chapter number 9, that is Biotechnology Principle and Processes of Class 12 NCERT. And in this session, in today's class, we are going to cover the last subsession of this 9.2 session, that is this whole session 9.2 was the tools for the recombinant DNA technology, and this last 9.2.2.3 we will complete, and then we will move to 9.3 session, which is the processes used in this recombinant biotechnology or recombinant DNA biotechnology things, and only cover the first subtopic, that is the isolation of the DNA. Okay, so let's start. Disclaimer. So what are going to be the learning objectives of our today's class? So in last two classes, first we have seen that how modern biotechnology have evolved so much by the combination of two engineering. First is the genetic engineering and second is the bioprocess engineering. Then we saw in last class that what are the tools which are responsible or which are useful during this, you know, during for this genetic engineering, and we have studied first the restriction endonucleases, which is the molecular scissor that is going to cut the DNA at the specific position, and we have talked about that thing in very detail in last class. The second was the cloning vector. So the what was the cloning vector? Which helped the foreign DNA to get inside the host and to replicate and makes it more multiple identical copies. So now we have studied about the two tools that we use in genetic engineering, but now it's time for third and the most important tool, that is host cell. Host cell is the cell in which we are going to transfer that recombinant DNA. If there is no host cell, there is no use of this recombinant DNA. We need some host cell to replicate those recombinant DNA and then produce whatever character or trait we want. So the first learning objective will be that we will explain the process of how we make this host cell competent, means how we make this host cell suitable for our DNA uptake, for our recombinant DNA uptake. Next will be we will see what are the different method of this DNA transfer, how we can transfer our recombinant DNA into host cell, what are the different methods. Then we will comprehend that how this host cell replicate foreign DNA, and along with that we also today going to start the how what are the processes of this recombinant DNA technology, and first step, that is the isolation of the DNA, that we are going to also talk about in today's class. So this is going to be the index for today's class. First we will complete this last subsession of 9.2, that is competent host, that and then we are going to move to the next session, that is the processes of recombinant DNA technology, and that which is the 9.3 session of NCERT. And in this we are just going to take 9.3.1 subsession, which is the isolation of genetic material, that is isolation of DNA. Then we are going to tell you the gist of all the slides and all the things we are going to cover in today's class. Then there will be some NCERT based question, and there are keynotes that you're going to take away with you guys. And then in last we are going to have some interesting questions that you guys gonna solve yourselves. So let's go. Competent cell. What is a competent cell? Competent host, sorry. So a competent host is a host which is suitable to take the foreign DNA inside it. And the definition says that a competent host for transformation with recombinant DNA is a cell that has been treated to become capable of taking up foreign DNA from its environment. This means a competent host is a cell which is suitable and which is capable of taking that foreign DNA, that is the recombinant DNA, into itself and get transformed. Because in last class we have studied what is transformation, the process by which the host cell going to take the recombinant DNA and transform itself. So this is a competent host sister cell which is suitable to take this foreign DNA, that is recombinant DNA, and become transformed. So what should be the common characteristic, what should be the common features of this competent host? First is that its cell wall or membrane should allow this DNA entry. A guest can get inside your home only when you open the door. So this host should allow this DNA to enter into itself. So it should be be modified in such a way that cell walls and membranes allow this DNA entry. And how it is achieved? So it can be achieved by some chemical or electrical treatment that we are going to study further and in more details in upcoming slides. So now you guys understand what is competent host. You can write in chat yes or no, or shall I go ahead? Okay, so I'm moving ahead. So this diagram is a pictorial representation of this transformed cell. This you're going to understand more clearly in upcoming slides, but I will give you a gist here also. So this is the E. coli cell, this is the host cell which is going to take the um, foreign on this recombinant DNA. We have done some chemical treatment, that is calcium chloride treatment, which we're gonna talk in detail in upcoming slides. And we see that this recombinant DNA got into this competent cell and completely became a transformed cell. Okay, so this is the characteristic of competent cell, and this is how a competent host becomes a transformed cell. So next. So what should be the properties of this competent cell? It should show very high transformation efficiency. Here high transformation efficiency means that if we are injecting, like if they are 20 cells, okay, so it should be like that ki in from 20 cell at least 50, 60, 70% cells should transform. So it efficiency should be very high. It should be genetically stable. It means that if we are even introducing the recombinant DNA it into it, it should be remain stable. It should show very rapid growth, because if it's showing very slow growth, then there is no point of like e coli divides in every 20 minutes. So if a cell get transformed, this host cell, this e coli get transformed, so in every 20 minutes you are getting your transformed cell. You understand? So this is very, it should be like that, it should show very rapid growth. Then it should be easily cultivated, means definitely we are going to make this recombinant, this transform cell into the cell in laboratories. So it should be very easy to culture. It's not like it's very difficult, and that it is not even possible, humanly possible to cultivate. It should be like easily cultivated, and it should going to have selection markers, because in the last class we have in detail have studied about selection markers. So if there won't be any selection markers, then we won't be able to, won't be possible for us to distinguish like which cell got transformed and which cell doesn't got transformed. Okay, so there should be a selection markers. So most commonly used competent host are E. coli, and many strains of E. coli has been used as this competent host. First is E. coli DH5 alpha. This is the most commonly used DH5 alpha. And if you guys even remember this DH5 alpha, it is more than enough. But others are JM109, BL21, TOP10. Here again, that picture only, we have given that pictorial representation only, that how that E. coli cell, that is a host cell, became competent cell and then became a transformed cell. And this method we are going to talk in later slides. So is there any confusion till now, that what is competent host, what should be their characteristic, what are their properties which make them a suitable host? So competent hosts again. Now question comes, how to make a host competent to take up DNA? First, the chemical method, that is the heat shock method. How we make a cell compet- a cell competent to take up the DNA? So first method is the chemical method. Also we call it calcium chloride method. Okay, because we use calcium chloride in this. So what we do in this, we treat bacterial cell with a specific concentration of divalent cation. You know divalent cation, a ion which have this 2 plus sign because they have lost 2 electrons. So this is the divalent cation such as calcium. What it will do? This increase the efficiency with which DNA enters the bacterium through pores in its cell wall. But the question arises, how this calcium chloride is able to, you know, how this calcium chloride makes this bacterial host as well as this DNA compatible, that it allow the entry. So there is one thing that you guys need to understand, that we know that DNA is negatively charged, okay, and it is polar, negatively charged and polar, whereas the cell membrane of, what is the composition of cell membrane? It is biphosphate it is lipid biphosphate layer, and we know that in lipid also it is like positive charge, it because they have polar head and non-polar tail and all those things. But you guys need to understand that this calcium chloride, it neutralizes the cell wall as well as the DNA and thus allow the entry by reducing the electrostatic repulsion between them. Because if there is negative charge there also and there also, then we know that that negative is going to repel each other. So it neutralizes both them and allow the entry inside it, and thus it increases the efficiency with which DNA enters the bacterium through pores in its cell wall. And how this happen and what is the procedure that we are going to study now. So what we do, first we incubate the cell with the recombinant DNA on ice. But we take this E. coli cells, we take the recombinant DNA and when we keep it on ice. Then we briefly place them at 42 degrees Celsius, and when we place them in this 42 degrees Celsius, now this is called heat shock. And why we give this heat shock? Because it is going to make pores on bacterial cell wall. Not sorry, cell wall, not it is going to make pores on this cell membrane, and that transient pores which they make during this heat shock, that allow the entry of foreign DNA into the cell. And then they put them back on ice. This enables the bacteria to take up the recombinant DNA. So this is like this is very superficial that how they do. We are going to talk about each and every step in next slide. So how they, as I told, this is also called calcium chloride treatment. Yeah. So now what they do? They take the E. coli, okay, and they grow this bacterial culture to mid-log phase. What is the mid-log phase? It is the exponential phase. Because we know that in exponential phase these bacteria divide rapidly and it is more receptive to take the DNA from outside. What is this OD 600 equals to 4, 0.4 and 0.6? You guys just need to understand that OD means optical density at the, so they have measured the optical density of this bacterial culture at the nanometer of 600, and this they usually do in microbiology and using spectrophotometer. They, it is basically tells us the what is the concentration of bacteria. So if at 600 nanometer, if they have taken the optical density and it came out to be 0.4 and 0.6, so we say that yeah, the culture have optimum number of bacterial concentration. So what they did, they grow the bacterial culture to this phase. They check it, through by measuring this concentration using spectrophotometer. They took it at 600 nanometer and find this optical density, which came out to be 0.4 to 0.6. Then they chill the cells on ice for 15 minutes. Why they do chill this cells on ice for 15 minutes? Because they want to slow down its metabolism. And now why they want to slow down its metabolism? Because in next step we are going to centrifuge it, and if we are going to centrifuge it, it may possible that the cell got disrupt or get destruct. So that's why they try to, slow down the metabolism of this bacteria bacterial culture. Now this centrifuge, I know you guys know that what is centrifuge. And in even last class I have even told that what is a centrifuge device. It is a device which, help to separate the materials on the basis of their density. So they centrifuge it at 4 degrees Celsius to pellet cell. So usually in a centrifuge, when we centrifuge something, what happens? Two things come. One is supernatant, which comes above, and then it's pellet. Here we want pellet because pellet is our cell and supernatant is the media. So we remove the media and we take this pellet cell. Then what we do now, because we have our E. coli, now we have our bacterial culture E. coli, now we resuspend this pellet in cold 0.1 molar of calcium chloride. Okay, so this is the that same representation, pictorial representation. So last step what they did, they took the cell and then that pellet, which is the cell, E. coli cells, now, and they suspend it in with the 0.1 molar of calcium chloride, and then they again incubate it, means keep it on ice for 30 minutes, centrifuge it again to, concentrate that cell and more, and then again resuspend in cold calcium chloride plus 15% should be there glycerol, because glycerol help in storage and all those things, and then liquate it and store it at minus 80 degrees Celsius. These steps, no one going to ask you these steps in exam or something, but you should have this just brief idea that how they perform it. Then now what happened, they have taken those E. coli cells, mixed it with calcium chloride, everything, and then store it in very high low degrees Celsius. Now it can, it can become freeze. Okay, it can freeze. So now they will take that cell which is in so low temperature and thaw this competent cells on ice, because it has already been treated with calcium chloride, so we can assume that it became competent. Thaw competent cell on ice means, thaw means they warm the cells at the 37 degrees Celsius. So now when this, they thaw it and bring it to the normal room temperature 37 degrees Celsius. Now they add this plasmid DNA to the cell. This plasmid DNA means recombinant DNA to the cell, and then again incubate on ice for 30 minutes, and then give it a heat shock at 42 degrees Celsius for 45 seconds. So when they are giving this heat shock, this is the time when this pores, so here is the pictorial representation. So this our cell. Okay. Now, and this is the plasmid, that is the recombinant DNA. Now, when we gave it heat shock for 42 degrees Celsius, it, you can see here, its cell membrane, you see, its it becomes what? It start making pores, transient pores, and from this pores we can able to, you know, it is able to go inside this recombinant DNA is able to get inside into this bacterium and thus became transformed. After this heat shock treatment, we return it to ice for 2 minutes. Then we do our normal things because now it became now it didn't being it became the transformed cell right right away only, because not every cell can become transformed cell now, but we have to culture it. So they add this media, then they incubate at 75 37 degrees Celsius at for like 1 hour. Then they plate it for selective media to see which cells became transformed, which cells transform, and which cells doesn't transform. So that is again the same pictorial representation. So this is like about, we have studied that how using the chemical method we can transform and make this host cell competent. Okay. So now we have talked about the chemical method. Now it's time to talk about the physical method. So first is the micro injection. Everyone has seen the injection, how from injection we can insert something into our body. Just like that, using a fine glass micropipette or a micro injection, we can inject this DNA directly into the nucleus of an animal cell. Okay, and the best thing about is this, that it offers very high precision, because you know where you are putting the your recombinant DNA. In chemical method, it was a hit and trial. Like some can go, some cells can transform and some cells cannot transform. But when we are using micro injection, it became like very high precisely that we are going to deliver the specific amount of DNA and at specific position into the nucleus of the animal cell. It is usually suitable for transforming the large DNA construct. Definitely it is a very, skill, like very precise job, so it requires very highly skilled person due to the delicate nature of this procedure. So not everyone can do this, only specific and people who are expert in this can perform this. So here you can see in the picture that this is the host cell, this is the nucleus of the host cell, and how using micro injection needle with foreign DNA, means in this we have our recombinant DNA, we are going to insert it into the nucleus. Other genes transfer method. So okay, so like for micro injection only, they have like told that how low throughput as only one cell can be injected at a time. Definitely, because at one time we can put injection into one cell only, so it will give low throughput. Secondly, it is commonly used in producing transgenic animals such as mice. Then it is applied in cloning and stem cell research. You guys know stem cell? What is stem cell? It is the undifferentiated cell. Okay, and this cell has the ability that it can differentiate into any cell. So we can use in this stem cell research also. Same picture is showing that how microinjection method of gene transfer take place. After this microinjection method, there is another method which is biolistic and the gene gun method. Here what we do, we use gold or tungsten particles and coat we take our DNA and then we coat it with our gold or tungsten particles, which are then shot into the cell using high pressure gas or electric pulse. First question arises, why we only use gold or tungsten particles? Why we can't use some other particle? So the first reason is that gold or tungsten particles are usually inert, that it is biologically inert, and it is not going to react with the host cell. That is the reason we are using this gold or tungsten particles. We coat this DNA with this gold or tungsten particles, and then this get shot into the cells. It get into the cells using the high pressure gas or electric pulse. There is a pictorial representation also here in the slide that I'll show you guys. For which organism it is useful? So it is mostly effective for plant cells with tough cell walls, and you know, in which we used to eliminate the needs for protoplast. It works on variety of cell types including bacteria, fungi and animal cells also. So here is the pictorial presentation. So you can see this is our target plant cell, okay, and this is the disc stopped by this no, this is not disc, this is this plastic disc. This disc contains DNA coated gold particles, okay. Now what will happen, this is a gene gun barrel, and what will happen, this high pressure gas or electric pulse will come from backward, and this will create a pressure on that, so it will start, it will start getting shot into this plant cell. So this is how it works. You do guys don't need to know the mechanism or something very of high level. You guys just need to know that this is one of the another method, ballistic and gene gun method, which is one of the method, physical method to make this competent cell. First, in this what happened, this DNA get covered with gold or tungsten particles, because gold or tungsten are inert, they are biologically inert, they are not going to react with cell. Now they get shot into the cell using this high pressure gas or some electric pulse. It usually effective for the plant cells because it is going to have the tough cell walls, okay, but we can use it for others also. But these things you guys need to know, that's gold and tungsten particle, high pressure gas or electric pulse, and yeah, just its name, gene gun, or it is also called ballistic. I'm moving ahead. There are some disadvantages to this. First, this very high velocity particle may damage the cell, and there could be some random DNA integration, and which can lead to the inconsistent gene expression. In this micro injection we can see that we are very precise that what we are doing, where we are injecting our recombinant DNA. But here, this random DNA integration can lead to inconsistent gene expression. It is usually widely used in crop genetic modification, such as in wheat and corn. This wheat corn examples we are going to study in next chapter. Yeah. So its application is that it's applied in chloroplast and mitochondrial transformation research. Here is this picture again showing that how you can, use this ballistic or gene gun. Another, this now we have studied chemical method, we have studied physical method, now it's time for biological method. So now for this biological method we can use disarmed pathogen. What is mean of pathogen? Pathogen, we know pathogen are the pathogen is something that cause disease. What is disarmed pathogen? Disarmed pathogen is the pathogen whose ability to cause disease has been altered, that it can go inside that thing which it is infecting earlier also, but now it cannot cause any disease, it got disarmed. For example, this agrobacterium tumefaciens that we have also talked in last class, it go and it has the ability to transfer this TI-DNA, and this TI-DNA is responsible for tumor development. But what we can do, we can disarm it tDNA and integrated with some foreign DNA which is useful or which we need to check into this plant, and thus utilize this modified pathogens, that is agrobacterium tumefaciens for plants, and retrovirus. Last class I have told that how retrovirus has the ability to convert the normal cells into cancer cells. Okay, but if we will disarm these pathogens, these can deliver the DNA into host cell without causing any diseases. It is highly efficient for certain hosts, such as dicot plants. This agrobacterium that is mostly efficient for this dicot plant only. It initially also it is used to uh, in induce this tumor in dicot plants only, and then it ensures the stable integration of DNA into the host genome, because it is biology. Its work is to last time to transfer the TI-DNA into that genome and cause this tumor and all those things. So it ensures that there is stable integration of our gene of interest into the host genome, because it is biologically made for that. So this has been shown that how this TI plasmid, we remove and replace these gene, the part which is responsible for causing this, uh, you know, tumor. We remove and replace these genes with our gene of interest, and thus we can disarm this pathogen and use it for gene transfer. So there will be some drawbacks for this also, biological method also. So what are those? First of all, it going to limit the host range, definitely, because we know that this agrobacterium is mostly for dicots, so it is not going to be that effective for monocots. Then there is some safety concern arise when using viral vectors in human gene therapy. As I told, this retrovirus, we its usual role is to that it converts the normal cell into the cancer cells, but we thought that okay, we have disarmed these pathogens, and we have inserted our gene of interest, and we then injected into a human. And you- in some gene therapy, where I told what is gene therapy, which help in treating the disease and all in last class. So if but we didn't know that it didn't work properly, so it and we injected into the human. What will happen? He will develop tumor, because this retrovirus real role is to convert this normal cell into oncogene cell, means tumor cell. So it creates some safety concerns also. It is used in developing some transgenic plants like soybeans and tomatoes. It's applied in This is its application. These two are its application. Former two were its disadvantages. It applied in gene therapy and vaccine research using viral vectors. Nowadays, a lot of viral vector have been used in gene therapy, and it is a very hot topic. This again shows that how disarm pathogen vectors method of gene transfers can be used. Now, what are the limitations of making competent cell? So what are the things which are making us limit our research and limit our scope of this competent host? First is host range specificity. For every vector, as I have told this, we last two classes we are discussing that how this foreign DNA go into the vector and how it make recombinant DNA and then it go into host. Every vector can be specific for this their host. Means it is like for E. coli, like E. coli is the host. Okay, it's not like that a vector which is working for E. coli, it's going work for yeast also. So its specificity is very low. Then size limitation for inserted DNA. It also depends, like E. coli cannot accommodate a very big or large DNA. It's like its DNA size should be less than 10 kilobase pair. For larger DNA we have, and even if we are inserting some larger DNAs into some host, we have to see whether they are even stable or not genetically. So these are the issue. Now, potential protein folding issues. So it also can happen, like E. coli. It has not that great machinery on to to tackle with the eukaryote protein foldings. So these protein folding issues can also arise. And post translation modification limitation. So we know that even after translation, that mRNA translate into protein, there are some modification can has to take place for like glycosylation, phosphorylation. Okay, you might have studied in earlier classes, or you might will study in some other classes. So these are the methods, these are the some modifications that need to be done in protein. But it's not possible for every host. Like E. coli cannot do it for it. Okay, like in therapeutic proteins, so there are some therapeutic proteins which need this glycosylation step, but it's not possible in E. coli. So there are a lot of limitations also, which like every cells can't be make competent host. Everything has its own pros and cons. Okay, so before that, now till now what we have studied, that what are the different methods by which we can make our competent cell, chemical, physical, biological, its disadvantages and all those things. Now it's time for to study the process flow of this, how it has been make. So what are the steps to make this recombinant DNA technology? First, because right till now we were studying about tools, so now it is like to know what are the steps. First is the isolation of the DNA. We need our desired DNA. Then we need to break that, fragment that DNA by restriction endonucleases, which we talked in last class. Then we have to isolate of that desired DNA fragment, which we talked in last class, like how we can take out our DNA fragment using gel electrophoresis. Then we will elute out our uh, desired DNA. Now, after taking our desired DNA fragment, we need to ligate it to the vector to make the recombinant DNA. After ligating using DNA ligase, we have to transfer this recombinant DNA into the host. And then we have to culture this host cell in a very large, in medium, at a large scale, and extract a desired product. Because this is what the ultimate goal is. We need to get the product. Okay. So this is the process flow overview, that how we first isolate our desired DNA, then how we will fragment that DNA by restriction in endonucleases, then how we will isolate this desired DNA, then how we will use like DNA ligase and ligate this DNA uh, fragment into vector, then how we will transfer our recombinant DNA into the host, and last how we will culture this host into a large, in medium, at large scale, and ultimately get the product which we wanted, our desired product. So the first step which we are going to talk about in today's class is isolation of this genetic material, that is DNA. So what is the objective of this process? That we want our pure DNA, that there would be no other macromolecules like RNA, proteins, polysaccharide, lipids, nothing, only our DNA. So how we can do? First we have to break the cell, that is what you call cell lysis, sorry. So the cell wall and or the membrane must be broken so that our DNA can release. So in case of bacteria, we use lysozyme, because we know that bacteria is made of cell wall is usually made up of peptidoglycan, and you know we this lysosome has the ability to break that. In plant cell, we know that its cell wall is made up of cellulose, therefore we need cellulase. In fungus, we know that its cell wall is made up of chitin, that's why we need chitinase. And this is the pictorial representation of how cell lysis happens. So this is the cell membrane, this is the nuclear membrane, this is the nucleic acid. We will lysis of cell wall or cell membrane happens, and then lysis of nuclear membrane happen, and this nuclear acid comes out from the nucleus. This is what we wanted at the last. So along with this in the cell, along with this DNA, there will be definitely RNA also. So how we will remove RNA? By treating it with the RNase, that is ribonuclease. Then how we are going to remove this protein? We will remove the protein by treating it with protease. You can see that similarity, that in every, like, which is able to break or cleave those things, are usually, like, last you write A's and A's. Protease means they are breaking or they remove this protein. RNA for ribonuclease. Chitin, it's chitinase. For cellulose, it is cellulase. Like this. And this is this how this last now, because we have removed everything. So now, like, last, this is also an NCERT picture. So at last you will get this, and this is what you call spooling. In this, you just cube out your DNA. So after this, we will remove this RNA, this protein, and all those things. Now what we will do, we will add we will add this chill ethanol into this solution, and by this what will happen, this purified DNA comes and precipitate out, and this precipitated DNA we can see as collection of fine threads, and this fine threads can be removed using the method called spooling, which you have seen in this last picture. This precipitated DNA can be removed from the suspension by winding it, swirling it onto a glass rod or spool, which we have shown in this figure also. This is a spool. Okay. So, and what it happened, all this DNA get winded in onto this glass rod or spool, what you call, and this is how we can isolate our DNA. So now, what is the summary of today's class? In short, first of all, how we started that what is the competent cell. Okay, that competent cell is the cell who has the certain characteristics that allow the entry of foreign DNA into itself. Okay, then we have studied what are the different methods. First we talked about chemical method, that is calcium chloride method, in which we get into deep process, like how first we treated with calcium chloride because it's going to neutralize the charge on DNA, and then how it, using heat shock method, we make small pores on this and this foreign DNA can get inside it. Then we talk about some physical method, that is the ballistic, gene gun, micro injections. Then we talk about some this biological methods by using disarmed pathogens, which has the ability to cause disease, but it has been disarmed. Then we talked about the processes, how we can make this recombinant DNA technology take place, what are the different steps. And after talking about those different steps, that how we isolate, what is the fragmentation and all those things, we talk how this isolation of DNA take place. Okay, how this take place, first we try to remove this cell wall and cell membrane, that in case of bacteria we use lysozyme, in case of plant we use cellulase, in case of fungus we use chitinase. Then we use, now after when we got the nucleic acid, protein and all those things, then we use ribonuclease and proteinase to remove RNA and protein. And then at last we use chilled ethanol to precipitate out our DNA, and we take out this DNA with the help of spooling, which we use with glass rod or spool, in which what happened, this DNA go and wind up around the spool, or what you call glass rod. So this is the summary, that organism capable of taking a foreign DNA, usually E. coli bacteria we used, and this I told now that strain, that TH alpha, it is used mostly. Then multiple gene can be inserted and can be expressed also, can be made competent through chemical or electrical treatment, or even biological, that we have studied. It provides cellular machinery for DNA replication and protein expression. The gene transfer methods include this physical method that we have already talked about, this calcium precipitation, this biological agrobacterium mediated transfer, then how this DNA isolation take place, that we involves breaking cells to release DNA, use enzymes like lysozymes, and detergent can be also used. Then we separate this protein and RNA. It's requires some purification step as well. So first question, what is the competent host in recombinant DNA technology and why it is essential? So because time is less, I am telling you answer. A competent host is a cell that can take up the foreign DNA and allow it to replicate. It is essential because it serves as a living factory for DNA replication. It provides all necessary cellular machinery for gene expression. Its common hosts include E. coli, yeast, or any mammalian cells also. It must be made artificially competent through chemical or physical treatment that we have talked about. It enables mass production of desired gene or proteins. Second question asks that what is the first step in recombinant DNA technology and how it is performed. So first step in the DNA recombinant technology, what it is, that's already we talked about is that only, that is the isolation of genetic material, that is DNA. How we extract the DNA is extracted from the donor organism using various methods, physical method, cell grinding, sonication, in which we use ultrasound waves to take our DNA, or pressure treatment, or there could be some chemical treatment like we talked about using enzymes, or we can even use chemicals like EDTA. The process involves cell lysis, that is removal of the cell wall and cell membrane, removal of protein and RNA. Then we this precipitation of DNA happen when we add this chilled ethanol. Then this DNA purification is the last step. Here are some keynotes. So first, DNA isolation was done by Friedrich Mischer in 1869. This natural transformation was discovered in 1928. This artificial competence method were developed in 1970s. Modern kits can isolate DNA in under 30 minutes. Some bacteria are natural competent. DNA quality affects transformation efficiency. Definitely it is going to affect that. Cell wall structure influence competence methods. Here are some interesting questions that you guys gonna solve yourself. That what is the significance of heat shock and bacterium transformation that we have talked in today's class. Explain why RNA contamination is undesirable in DNA isolation. That also you guys have to find out and tell. We have given some hints in these slides also. What factors affect transformation efficiency when generating competent cells? That also, so we have given some hints even in the last slide you can find. So yeah, last question, how does ethanol precipitation helps in purifying DNA during RN this isolation process? So as I have told, like when you will add this ethanol, it will precipitate out. How and why, that you guys find yourself. Otherwise, you guys can ask me in tomorrow's class. Okay. Thank you. I will open this TPP question. So here's the TPP questions. So what the first question says, that why is calcium chloride treatment used to make bacterial cells competent for recombinant DNA transformation? That why this calcium chloride treatment is used and why it is able to make this bacterial cell competent for recombinant DNA transformation. So let's see what this says in option. First, it neutralizes the negative charges on DNA and cell membranes, reducing electrostatic repulsion. Yes, we have uh, studied in the chapter this class only that how this calcium chloride, which is mainly the calcium, this divalent cation, neutralizes the negative charge on DNA and cell membrane and reduce the electrostatic repulsion. But let's see other options also. Okay, it degrades the bacterial cell wall, allowing DNA to enter easily. No. It increases the hydrophobicity of DNA, enabling membrane penetration. No. It acts as a nutrient source for bacterial growth. No. So the answer would be first, it neutralizes the negative charge on DNA and cell membrane, reducing electrostatic repulsion. Now, what second question says? It says, what is the purpose of the heat shock step 42 degrees Celsius in bacterial transformation? Like why we do this heat shock step in during this bacterial transformation when we are using this calcium chloride steps. So let's see the options. First, to kill competing non-transform bacteria. No, we don't do that. Don't do it for that. Second, to create temporary pores in the bacterial cell membrane for DNA uptake. Yes, we have talked in the class only that how this heat shock helps in making this transient pore on the cell membrane for DNA uptake. So second option. But let's see the other options too. That third option is to activate DNA polymerase for recombination for replication of recombinant DNA. No, it has nothing to do with this DNA polymerase activation or anything. To denature the plasmid DNA for integration into the genome. Obviously not, because if we will denature the plasmid DNA, what is the benefit of all this transformation step? So the answer would be to create temporary pores in the bacterial cell membrane for DNA uptake. So we will save. And next, which of the following method, the third question says, which of the following method is not used for introducing recombinant DNA into plant cells? So they are asking which is not this method for transferring this recombinant DNA into plant cell. Here, pay attention, plant cell, because all of this option which I am seeing is this method only. What do you say? For transferring recombinant DNA only, but here question specified that which of this following method is not used for introducing recombinant DNA into plant cell. Okay. First is ballistic. Ballistic, yeah, we can use. Okay. Ballistic, we can use. Microinjection in plant cells, yes, we can use. Agrobacterium mediated transformation, we know that this agrobacterium is going to inject into dicot plants only. Fourth, calcium chloride treatment, no, this can't be, we haven't studied it for plant cells. So the answer would be calcium chloride treatment. Now we will save it. And next, in ballistic method, in this gene gun method, the DNA to be delivered is coated onto microparticles of this we have again studied in the class that, but let's see the option. Silver or platinum? Nope. Gold or tungsten? Yes. And this other iron or nickel, copper or zinc? No. Only gold or tungsten. Why? Because it is biologically inert and it is not going to react with cells. That's why gold or tungsten. Now, the last question, which says, which statement about microinjection is correct? Which statement among these is correct about microinjection? First statement says, it is used exclusively for bacterial transformation. Is it so? I don't think so. Second, recombinant DNA is injected directly into the nucleus of an animal cell. Yeah. Okay, but let's see other options also. It relies on heat shock to facilitate this DNA uptake. We know that this DNA shock, this heat shock method step is used in only this chemical method, that is calcium chloride treatment. Now, fourth one, it involves the use of disarmed viral vectors. No, this is not used in disarmed viral vector because it's itself a biological method for this making this competent host. So the answer would be second, that is recombinant DNA is injected directly into the nucleus of an animal cells. So now let's see what will be the answer. We will save this and see, we will submit our answers. All answers are correct. So yeah. So we are done for today's class. Let's meet in next class.